So, so the respiration in plants, chapter 12. Now, context are: do plants breathe? Glycolysis, fermentation, aerobic respiration, the respiratory balance sheet, amphibolic pathway, and respiratory quotient. Now, all living organisms need energy for carrying out daily life activities. Okay. Uh, all the energy required for life processes is obtained by the oxidation of some macromolecules that we call food. Green plants as cyanobacteria can prepare their own food by the process of photosynthesis. They trap light energy and convert it into chemical energy that is stored in the bonds of carbohydrates like glucose, sucrose, and starch. Glucose, sucrose, and starch. Okay, we must remember that in green plants too, not all cells, tissues, organs photosynthesize. Only cells containing chloroplasts that are most often located in the superficial layers carry out photosynthesis. Okay, the food has to be translocated because other organs, tissues, cells that are non-green need food for oxidation. Animals are heterotrophic, that is, they obtain food from plants directly if they are herbivores, or indirectly if they are carnivorous like us, like humans. Saprophytes like fungi are dependent on dead and decaying matter. Okay, so ultimately all the food that is required for life processes comes from the photosynthesis. This chapter deals with cellular respiration, or the mechanism of breakdown of food materials within the cell to release energy and the trapping of this energy for synthesis of ATP. So whenever we want it, we can use this currency, that is ATP. Now photosynthesis of course takes place within the chloroplast in the eukaryotes. Plants are eukaryotes, whereas the breakdown of complex molecules to yield energy takes place in the cytoplasm and in the mitochondria. Okay, as only in eukaryotes. The breakdown in mitochondria only in eukaryotes. The breaking of CC bonds of complex compounds through oxidation within the cells, leading to release of considerable amount of energy, is called respiration. The compounds that are oxidized during this process are known as respiratory substrates. Usually carbohydrates are oxidized to release energy, but proteins, fats, and even organic acids can be used as respiratory substances in some plants under certain conditions. Now, during oxidation within a cell, all the energy contained in respiratory substrates is not released free into the cell or in a single step. It is released in a series of slow stepwise reactions controlled by enzymes, and it is trapped as chemical energy in the form of ATP. Hence, it is important to understand that the energy released by oxidation in respiration is not, or rather cannot be, used directly, but it is used to synthesize ATP, which is broken down whenever and whatever and wherever energy needs to be utilized. I told you before. Hence ATP acts as the energy currency of the cell. Same, this energy trap in ATP is utilized in various energy recovering process of the organisms. Okay, now do plants breathe? Yes, they do. Plants require O2 for respiration to occur. They also give out CO2. Okay, hence plants have systems in place that ensures availability of O2. Now plants, unlike animals, have no specialized organs for gaseous exchange, but they have stomata and lenticels for this purpose, right? Now the reasons why plants can get along without respiratory organs. First, each part taking care of its own gaseous exchange needs, and there is very little transport of gases from one plant part to another. Okay. Second, plants do not present great demands like humans for gas exchange. Roots, stems, and leaves respire at rates far lower than animals do. Okay. Only during only during photosynthesis are large volumes of gases exchanged, and each leaf is well adapted to take care of its own needs during these periods. So when cells photosynthesize, availability of O2 is not a problem in these cells, since O2 is released within the cell from the H2O at the PS2 P680 nanometer photosystem 2. Okay, third, the distance that gases must diffuse even in large bulky plants is not great. Each living cell in a plant is located quite close to the surface of the plant, so This is true for leaves. Now you might think of, what about thick woody stems and roots that inside? In stems, the living cells are organized in thin layers inside and beneath the bark. They also have openings called lenticels. Now these living cells have the openings called the lenticels. The cells in the interior are dead and provide only mechanical support. Thus, most cells of a plant have at least a part of their surface in contact with air. This is also facilitated by the loose packing of parenchyma cells in leaves. So this is also facilitated by the loose packing of parenchyma cells in leaves, stems and roots, which provide an interconnected network of air spaces. Okay, loose packing of parenchyma cells that provides the interconnected network of air spaces. Now the complete composition of glucose, which produces CO2 and H2O. So combustion gives carbon dioxide and water as end products, yields energy, most of which is given out as heat. Now C6H12O6 plus oxygen, this is 6 oxygen, this going to composition making 6 CO2 plus 6 H2O plus energy. Now if this energy is to be useful to the cell, it should be able to utilize it to synthesize other molecules that the cell requires. The strategy that the plant cell uses is to catabolize the glucose molecule in such a way that not all the liberated energy goes out at haste. The key is to oxidize glucose not in a one step but in several small steps, enabling some steps to be just large enough such that the energy released can be coupled. The step should be large enough so that the energy can be coupled to ATP synthesis. During the process of respiration, oxygen is utilized and carbon dioxide, water and energy are released as products. During the process of respiration, oxygen is utilized and carbon dioxide, water and energy is released as products. The combustion reaction requires oxygen, right? But some cells live where oxygen may or may not be available. Can you think of such situations and organisms where O2 is not available? There are sufficient reasons to believe that the first cells on this planet lived in an atmosphere that lacked oxygen. Even among present-day living organisms, we know of several that are adapted to anaerobic condition. So some are adapted to anaerobic condition. Some of these organisms are facultative anaerobes, that is, they are optional anaerobes, while in others the requirement for anaerobic condition is obligate. So some are facultative anaerobes, some are obligate anaerobes, that is compulsory, can only survive without the oxygen. In any case, all living organisms, in any case, be it anaerobic, be it aerobic, in any case, in any type of organisms, all living organisms retain the enzymatic machinery to partially oxidize glucose without the help of oxygen. Okay, this breakdown of glucose to pyruvic acid is called glycolysis. Now talking about the glycolysis. What is glycolysis? The term glycolysis has originated from Greek words glykos for sugar and lysis for splitting. So it is the splitting of sugar, means splitting of sucrose, means splitting of carbohydrates. The scheme of glycolysis was given by Gustav Embedded Meyerhoff and J. Parnas. So it is often referred to as EMP pathway. In anaerobic organisms, it is the only process in respiration. Glycolysis occurs in the cytoplasm of the cell and is present in all living organisms, right? In this process, glucose undergoes partial oxidation from two molecules of pyruvic acid. In plants, this glucose is derived from sucrose. In plants, the glucose is derived from sucrose, right, which is the end product of photosynthesis. In plants, in this process, glucose undergoes partial oxidation from two molecules of pyruvic acid. In plants, this glucose is derived from sucrose, which is the end product of sucrose, which is the end. So sucrose is the end product of photosynthesis, and this glucose is derived from it to undergo the partial oxidation from the two molecules of pyruvic acid during the glycolysis process, or from storage carbohydrates, right? Now sucrose is converted into glucose and fructose by the enzyme invertase, and these two monosaccharides readily enter the glycolytic pathway. So sucrose gets converted into glucose and fructose by the enzyme invertase. Okay, now glucose and fructose are phosphorylated. the phosphorus is getting attached to give rise to glucose 6-phosphate and fructose 6-phosphate by the activity of the enzyme hexokinase. Okay, now this phosphorylated form of glucose then isomerizes to produce fructose 6-phosphate. So glucose 6-phosphate gets converted into fructose 6-phosphate, and if it is fructose, then it gets converted into fructose 6-phosphate. Okay, so this phosphorylated form of glucose then isomerizes to produce 6-fructose 6-phosphate, right? Subsequent steps of metabolism of glucose and fructose are same. Now, in glycolysis, a chain of 10 reactions under the control of different enzymes. 10 reactions and 10 different enzymes takes place to produce pyruvate from glucose. So there is utilization or synthesis of ATP or NADH plus H plus takes place during the glycolysis. Now ATP is utilized at two steps. First, in the conversion of glucose into glucose 6-phosphate. So sucrose gets converted into glucose and fructose by the invertase enzyme. Then glucose is converted into glucose 6-phosphate by the enzyme hexokinase phosphorylated. Now when the glucose is converted into glucose 6-phosphate, there is one ATP is released, one ATP is utilized, because from the phosphorus from ATP it gets attached to the glucose 6-phosphate in. So this is why. And second, in the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate, as you can see in the diagram, that is fructose 6-phosphate. So the next step, okay, glucose into glucose 6-phosphate, ATP is released, ATP is used, then glucose 6-phosphate to fructose 6-phosphate, that is isomerases. Okay, now fructose 6-phosphate to fructose 1,6-bisphosphate, another phosphorus is added, so ATP is utilized. So this is how it does. Okay, now the fructose 1,6-bisphosphate is split into dihydroxyacetone phosphate and 3-phosphoglyceraldehyde PGAL. Okay, so 3 carbon. Okay, we find that there is now similarly there is one step where NADH plus H plus is formed from NAD plus. Now this is when 3-phosphoglyceraldehyde, that is 3 PGAL is converted into 1,3-bisphosphoglycerate BPGA. So that's it. Like one glyceraldehyde, 3-phosphoglyceraldehyde gets converted into 1,3-bisphosphoglyceric acid. So in which NADH plus H plus is formed. Now we find, okay, 2 redox equivalents are removed from PGAL, that 3-phosphoglyceric acid, and transferred to a molecule of NAD plus. Now PGAL, that is 3-phosphoglyceraldehyde or glyceraldehyde 3-phosphate is oxidized and with inorganic phosphate to get converted into BPGA. This is oxidized, it gives the hydrogen, it gives the redox equivalents in the form of 2 hydrogens, and it gets itself oxidized and with inorganic phosphate to get converted into BPGA, that is bisphospho 1,3-bisphosphoglyceric acid, right? Now the conversion of BPGA, that is 1,3-bisphosphoglyceric acid to 3-phosphoglyceric acid PGA, is also an energy yielding process. It gives an ATP, okay, because one phosphate is getting removed in to make the ATP. Now this energy is trapped by the formation of ATP. Another ATP is synthesized during the conversion of PEP, phosphoenol pyruvate, to pyruvic acid in the last step. Okay, and before that last step, when 2-phosphoglycerate is converted into phosphoenol pyruvate, water is released. Okay, now can you then calculate how many ATP molecules are directly synthesized in this pathway from one glucose molecule? So how many ATPs are directly synthesized? It's 4 ATPs are directly synthesized, because 2 molecules we are talking. First in the glucose to glucose 6-phosphate, then second when glucose 6-phosphate to fructose 6-phosphate, that isomerizes, that is hexokinase. Then fructose 6-phosphate to fructose 1,6-phosphate, there is another ATP is formed, utilized, but when it is formed, when 1,3 bisphosphoglyceric acid to 3 PJ, that is 3 phosphoglyceric acid. So what, 2 molecules, so 2 ATPs is formed, and another phosphenol pyruvate to pyruvic acid, that is 2 molecules, so 2 ATPs is formed. So total 4 ATPs are formed. Okay, now pyruvic acid is then the key product of glycolysis. What is the metabolic fate of pyruvate? This depends on the cellular need, oxygen, and the type of organism. Now, there are three major ways in which different cells handle pyruvic acid produced by glycolysis. These are lactic acid fermentation, alcoholic fermentation, and anaerobic respiration. Okay. Now, fermentation takes place under anaerobic condition in many prokaryotes, unicellular eukaryotes, and the germinating seeds, that is given in the summary. Okay. Now, for the complete oxidation of glucose to CO2 and H2O. This is the complete oxidation. However, organisms adopt Krebs cycle, which is also called as aerobic respiration. This requires oxygen supply. So in the Krebs cycle, which happens in the mitochondrial matrix, this requires the oxygen supply. Now, fermentation. We are talking about the fermentation. So in fermentation, say by yeast, the incomplete oxidation of glucose is achieved under anaerobic conditions by series of reactions where pyruvic acid is converted to carbon dioxide and ethanol. In yeast fermentation, pyruvic acid is converted to carbon dioxide and ethanol. Okay, the enzymes pyruvic acid decarboxylase mean because CO2 is released, and alcohol dehydrogenase because it gets converted into aldehyde and then ethanol. So alcohol dehydrogenase catalyzes this reaction. So yeast fermentation, pyruvic acid is converted to CO2 and ethanol, then pyruvic acid decarboxylase because CO2 is released, and then alcohol dehydrogenase because the formation of ethanol. The other organisms like bacteria produce lactic acid from pyruvic acid. Now other bacteria as well, they produce lactic acid from. The steps involved are shown in animal cells also, like muscles during exercise. When oxygen is inadequate for cellular respiration, pyruvic acid is reduced. Pyruvic acid is reduced to lactic acid by lactate dehydrogenase. So pyruvic acid is reduced to lactic acid by lactate dehydrogenase in human muscles, like in human muscles. The reducing agent is NADH plus H plus, which is reoxidized to NAD plus in both the process. So reducing agent is NADH plus H plus. Okay. So it reduces pyruvic acid to ethanol. Okay, and itself get oxidized, that is NAD plus in both the process. Now in both lactic acid and alcohol fermentation, not much energy is released. Less than 7% of the energy in glucose. Less than 7% of the energy in glucose is released, and not all of it is trapped as high energy bonds of ATP. Also, the processes are hazardous. Either acid or alcohol is produced. What is the net ATPs that is synthesized when one molecule of glucose is fermented to alcohol or lactic acid? So what is the net ATPs that is synthesized? No ATP is being synthesized during the fermentation. Calculate how many ATP are synthesized and deduct the number of ATP utilized during. So no ATP is being utilized, and deduct the number of ATP utilized during glycolysis. 4 ATPs are formed during glycolysis and 2 being used, so net gain is 2 when one molecule of glucose is fermented to alcohol or lactic acid. Okay. Now yeast poison themselves to death when the concentration of alcohol reaches about 30%. What then would be the maximum concentration of alcohol in beverages that are naturally fermented? How do you think alcoholic beverages of alcohol contain greater than these concentrations are obtained? What then is the process by which organisms can carry out complete oxidation of glucose and extract the energy stored to synthesize a large number of ATP molecules needed for cellular metabolism? In eukaryotes, these steps takes place within the mitochondria, and this requires oxygen. Okay, aerobic respiration is the process that leads to a complete oxidation of organic substances in the presence of oxygen and release of CO2, water, and a large amount of energy present in the substrate. This type of respiration is most common in higher organisms. Now we are talking about the aerobic respiration. So for aerobic respiration to take place within the mitochondria, the final product of glycolysis, pyruvate, is transported from the cytoplasm into the mitochondria. The crucial events in the aerobic respiration are the complete oxidation of pyruvate by the stepwise removal of all the hydrogen atoms, leaving 3 molecules of CO2. The complete oxidation of pyruvate by the stepwise removal of all the hydrogen atoms, leaving the 3 molecules of CO2. So this one is the first key. The second one is the passing on of the electrons removed as the part of the hydrogen atoms to molecular O2 with simultaneous synthesis of ATP. The passing on of the electrons removed as a part of the hydrogen atoms to molecular O2 with simultaneous synthesis of ATP. So this is the two key features. Like, what is interesting to know that the first process takes place in the matrix of the mitochondria, that is the Krebs cycle, where the oxidation of pyruvate takes place by stepwise removal of all the hydrogens, leaving 3 molecules of O2. So this is the Krebs cycle which happens in the matrix of the mitochondria, while the second process is located on the inner membrane of the mitochondria, not the intermembrane but inner membrane. Okay, pyruvate, which is formed by the glycolytic catabolism of carbohydrates in the cytosol, that is cytoplasm, after it enters mitochondrial matrix, undergoes oxidative decarboxylation by a complex set of reactions catalyzed by pyruvic dehydrogenase. You see, we study the lactate dehydrogenase, we study the alcohol dehydrogenase, we study the pyruvic decarboxylase. You see in this paragraph, alcohol dehydrogenase. Okay, so in here it is a pyruvate dehydrogenase. So lactate dehydrogenase is different, pyruvate dehydrogenase is different. Now the reactions catalyzed by pyruvate now the reactions catalyzed by pyruvic dehydrogenase require the participation of several coenzymes including NAD plus and coenzyme. So pyruvic acid plus coenzyme A plus NAD plus, in presence of magnesium, pyruvic dehydrogenase acetyl coenzyme A plus CO2 plus NADH plus H plus. So during this process 2 molecules of NADH are produced from the metabolism of 2 molecules of pyruvic acid produced from the 1 glucose molecule during glycolysis. During this process 2 molecules of NADH are produced from the metabolism of 2 molecules of pyruvic acid produced from 1 molecule glucose during glycolysis. Because there are 2 molecules of pyruvic acid, so when it undergoes pyruvate dehydrogenase, 1 CO2 is 1 NADH is obtained, but it's 2 molecules, so it is 2. Now tricarboxylic acid cycle, that is citric acid cycle.